Hello there again, everybody. So here is our second of two lessons, just talking about mitosis. So what I want to talk about this time is something called the cell cycle. So this is essentially, in a nutshell, is the whole life cycle of any given cell. So it can be summed up in four different phases, technically speaking. So the first three phases is what you've identified, again, from the terminologies in your notes, as what's called interphase. Interphase technically can actually be broken down into three different steps. So we have the G1, or the G actually stands for gap. So they call it gap one phase. And then after gap one or G1 is finished, it moves into the S phase. The S, technically speaking, stands for synthesize. To synthesize means to make or to create. And what are you making or creating in this synthesis phase? You are making or creating additional DNA. Thus, it's in the parentheses down here, DNA replication. You're replicating your DNA. Well, that would make sense. You're synthesizing it to make or to create. After it's done replicating the DNA, it's going to move on to G2, so gap 2 phase. And after this gap 2 phase, it's going to move into this fourth and final phase, which you're going to be spending a majority of our time on here today, which is called M phase. The M stands for mitosis, or nuclear division. And at the end, to kind of wrap things up, technically speaking, we can tack it on at the end, which is called cytokinesis, or cytoplasmic division, to finalize one cell becoming two. So this is, again, in a nutshell, the entire life cycle of a cell represented in circular format. So G1, gap one phase, moving into the S, which is the synthesis phase, which means to make or to create, which you're making or creating additional DNA, thus DNA replication, and move on to G2 or gap two. After it's finished with gap two, moves on to the M phase, the mitosis phase. Okay, so some interesting things about this interphase, G1, S, G2, gap one, synthesize, and gap number two. Really, let me break these steps down. And I got a fun little acronym, I guess, a little sorts here. So the G1 or the gap one phase is what I like to call the grow phase. Or in the image in the upper right-hand corner over here, it says G1 phase cell growth. So the G, I like to think of it as, oh, this is cells growing. Okay, so G1, that's growth, cell growth. After that, it moves into the S. As we've already said, that's the synthesize phase. And to synthesize means to make or to create something. What are you making or creating? Additional DNA. Well, of course, ultimately, if you're going to end up with two cells, they need to be perfectly identical to each other, which means at some point you have to copy or replicate that DNA. Thus, this needs to be part of our interphase right here. We need to copy it before we can replicate the entire cells themselves. Then we're moving on to the G2 phase, which is the go phase. You're getting ready to go. You're getting ready to do some mitosis. You're getting ready to go ahead and move from one cell and divide perfectly even into two cells. Something interesting about these three phases is approximately, I mean, give or take, depending on the cells, it equates to about 90% of a cell's life. That's a lot of lifetime. And to put things into perspective, on average, we'll take a skin cell. I heard this stat here sometime in college, I want to say, that your skin cells, they go through this whole cycle right here in about 20 hours. Every 20 hours, that's less than a day, just slightly. Every less than 20 or about 20 hours, less than a day, your cells replicate. You go from one to two, approximately, again, give or take 20 hours on average. And 90% of that time, so about what, 18, 19 hours, somewhere around there, it just does normal cell growth activity. It's going to replicate the DNA, and then it's going to get ready to go to move into this M, this mitosis phase. So here we're going to break down our mitosis or M phase that it's sometimes referred to as. So this, oops, got cut off a little bit. This is a multi-step process, which we're going to be duplicating cells, like the image down here is showing. We're going from one cell to two cells, mitosis. So let's go ahead and jump straight into it and break it down. So first off, we have prophase. So I like to think of it, if you are a pro, you are number one, right? So you think of pros, these are the athletes, these are the fantastic people, these guys are number one. So I like to think of pro, number one. This is our first phase, this is our first up to bat. So first, what's going on in this prophase? Point number one, the DNA condenses. To condense really just means it's going to thicken. So all that DNA is going to wind up in the chromatin and ultimately produce these chromosomes. Look at, there's those X-looking structures again. So this is going to condense to form these chromosomes. And if you notice very carefully, you can see there's kind of a dashed line. There's like a dotted line going on here in this prophase. This is showing the nucleus right here, or this nuclear envelope, what's protecting the DNA, is slowly disappearing. So this is what's going on in our prophase. A few other things just to point out, and I'll just point them out once, and all the other slides, we'll just put them in the images here. So first off, we have centrioles. This, again, another one of our terms. Centrioles, these are these paired looking structures, kind of look like little cylinders here. And you'll see that there's two pairs of them. Here's one pair of centrioles, here's another pair of centrioles. These small paired structures which assist in the division of our cells as they go through this mitosis phase. Next, you can see these little thread looking things, or kind of looks like a crab or a spider or something. So you got these little threads coming down. We call these spindle fibers. These are string-like structures which then ultimately attach themselves to the centromeres. Here's another one of our buzzwords. They attach themselves to the centromeres so that later on down the road they can rip them apart. So prophase, DNA condenses or thickens to form our chromosomes. The nucleus, the nuclear envelope, starts to disappear. Your centrioles, these are the paired structures on the end, those are the green things, start to produce these little spindle fibers. The little fibers start to come out and move into our next phase. Okay, so the next one is what's called the metaphase. And what I like to think of, I got a little trick for this one, is, here's my trick. So for the metaphase, the DNA meets in the middle for metaphase. It's kind of got that M thing going on, right? Meets in the middle at metaphase. And if you notice in this image right here, we have all of our chromosomes, those X-looking structures, are meeting in the middle. They're all lining up and they're getting ready to get divided. So these spindle fibers, if you notice, they elongate or they start to grow and grow and grow, specifically from those centrioles. So these spindle fibers get long and ultimately attach to the DNA at the centromere location, right in the center, if you notice. That's why they call it centromere, centromere, right? So the spindle fibers are attaching and getting ready to move on to our next phase, anaphase. So after they attach themselves to the centromeres, eventually what they do, they are going to retract. So these centromeres are they're going to pull it back. When I like to think of this, think of it like almost like fishing, right? You cast your line, that's the spindle fiber. You cast it out, you hook your fish, it'd be like the chromosome here, and then eventually you got to reel it in. So these centrioles, they're reeling in using those spindle fibers, and they're going to pull apart the DNA and start to separate them. So the DNA starts to separate from each other. Now, I was told this little trick here from my AP biology teacher when I was in high school. So I don't know if this is true or not, but he always said, oh, I had a sister, Anna. Me and Anna, we don't get along. We always fight, and we always like to get away from each other. So he always said, oh, I need to get away from my sister, Anna. I don't know, maybe you do have a sister, Anna, maybe not. I don't know, it's a little trick. Maybe it'll help you, maybe it won't. So maybe you think, oh, I have a sister, Anna. Oh, we fight all the time. We got to get away from each other. So you got to get away or separate or get apart from each other during anaphase. So next phase we have what's called telophase, not telophase. Telophase is how it is. And you'll notice each of the new cells are starting to take shape. Also, what you notice is that nuclear envelope is starting to reform. So that nucleus is starting to reform to protect the DNA again. And you'll notice, again, these cells are further dividing to separate just a little bit further from another and getting ready to do our final phase, which is our cytokinesis. So in this phase, give all our clicks here, they do it slightly different between animal and plant cells here. So cytokinesis, the key here is that the cytoplasm and all the other additional organelles that are inside the cells here are then going to separate from one another and further divide, ultimately ending up with two separate cells. So animal cells, they have that plasma membrane, which is specifically going to be divided. It's going to sort of cleave or pinch off, versus a cell wall, it forms what's called a cell plate. So now that cell wall has to be reformed so it doesn't pinch and split like an animal cell might. It actually just kind of rebuilds the wall. Last but not least, here is our overview, just to kind of put everything into perspective over here. So we start off